Okay, students, so welcome back. Okay, so before continuing where we left, as I mentioned you when the Nandini was presenting, she talked about something grooves, and I mentioned you that these grooves are a bit three-dimensional things. So we can't understand really in the two-dimension what really this groove mean. So I'm sharing you some videos. So two videos are here. So these videos are going to explain you in three-dimensional structure way what these really grooves are and how these grooves are formed. Okay, so I'm sharing you this video. Please watch this video and listen these videos carefully. And my voice might not work when I'm sharing to you because there is going to be some issue in Apple.
The major and minor grooves of DNA are highlighted.
Okay.
Nucleobase atoms facing the major groove are colored pink, and nucleobase atoms facing the minor groove are colored blue. It's easy to see that the major groove is larger than the minor groove, but in this lesson we will look more closely at the structure to get a more precise definition of the major and minor grooves. If we focus in on the deoxyribose of the backbone, we can see the important relationship that defines the major and minor grooves. Looking at the left backbone, we can see that in the major groove, the deoxyribose always points its ring oxygen up. If you imagine the deoxyribose as the Statue of Liberty with the phosphate as its torch, you'll always remember the correct orientation for the left side of a major groove. Looking again at the left backbone, we can see that in the minor groove, the deoxyribose always points its ring oxygen down, kind of like an upside-down Statue of Liberty. This orientation makes sense because the two DNA backbones run in opposite directions. Always keep in mind the 5' to 3' directionality when viewing polynucleotides. Looking at the right backbone, we can now see that in the major groove, the deoxyribose always points its ring oxygen down. This is the opposite orientation compared with the complementary left backbone. And looking again in the right backbone, we can now see that in the minor groove, the deoxyribose always points its ring oxygen up. Again, this is the opposite orientation compared with the complementary left backbone.
And this is second video which will show you.
We now know that the twist angle between two adjacent base pairs will lead to the formation of the DNA helical structure. I want to spend a little bit more time on this because I want you to have a feeling of the three-dimensional structure of DNA here in this model of the DNA. We are looking from the top. Rectangular shapes represent nucleobases. We can draw the inner circle here with minor and major grooves. Using a computer modeling program such as Chimera is a good way to start. But to have a good feeling of the 3D structure, you might want to build your own DNA model. Here I made one, quite simple, from cardboard. This inner circle containing the bases with the center here. And I cut it into 2 pieces along the minor groove lines here. I'm going to use it for the model. You can make 10 of these, make hole at the center, and then put them into a metal wire as an axis. Now from the top, you can see that the top circle and the next one, the top circle and the next one have an offset of about 36 degrees, which, as we know, is good for the pi stacking. Note that the rotation here is clockwise, which will result in the right-handed DNA helix. We'll talk more about it later. So this side here is a minor groove side. You can follow it from the top to the bottom. The other side, this side right here, it's a major groove. You can see it follow here, and this is a major groove showing here. Major groove, minor groove. Now what I want to do next is to talk more about the characteristic of these two grooves that is important for the recognition of enzymes or a transcription factor that recognize DNA. Now imagine that you are an enzyme that can recognize a specific sequence of DNA, since the phosphodiester outside as well as the deoxyribose are quite similar in most cases. You have to rely on the patterns of the nucleobases in the inner circle showing here. More specifically, you have to rely on the minor and the major grooves because those are the site of nucleobases that are exposed, hence you can reach out to. What are the patterns here? Let's start from AT there. From the major group, we have acceptor, donor, acceptor, and here we have methyl group. Let's call it M. On the minor group, we have acceptor. Here we have H, and we have acceptor. Now let's move on to the GC pair. We have acceptor, acceptor, donor, and then we have H. On the minor group side, we have acceptor, donor, acceptor. Now, if you are an enzyme that recognizes DNA, you're going to read AD from the major group as ADAM. On the other hand, DA as MADA, which is different from, of course, AT. On the other hand, when you read GC, you're going to read as AADH and CG as ADAA, which occurs different from each other and also different from AT or TA pair. That's the major groove site. Now from minor groove, you're going to see the symmetry. AT is going to be AHA, and same as TA with it also AHA. Same thing here in GC is ADA, or CG is also ADA. So most enzymes gonna exploit major groups to recognize the specific sequence, while the minor group gonna apply in general cases for enzyme DNA interaction.
My name is Ariel, and I just started my first.
Hope these two videos explain you what really these grooves are and how these grooves are forming. Is it? Do you got the point? What these grooves mean? Because generally, what happens just we talk. So.
A very good evening to all of you. And
because generally what happens, we always talk in 2-dimension thing. So we generally rarely see DNA in the 3-dimension, because these grooves are the property of 3-dimension, and that 3-dimensional thing, how we are seeing in the 2-dimension that we are representing. So what face is in front of you when the face is minor one, means a minor one face is in front of you. So the minor face we are going to see for a shorter length, major face we will see for a longer length. So that's why we are saying that this is a in the length wise we are showing that this is a major groove and it is the minor grooves. So but in principle grooves are just the face of your nitrogenous base. So one face, one part of the phase nitrogen we are calling, it is going to correspond to the major one, and other one is going to correspond to the minor one, and they are rotating. So that's why you see in a way that image, that video that showed you how we are going to count it. Okay, so as she mentioned, how in what role they are playing role in the interaction with different enzyme, that how they are going to access to this hydrogen bond, so they can open it up and they are going to close it up. Okay, so this is about the year one. So now we can continue with our quick run, a quick rush through the slides. Okay, so now we are back to our slide mode. So you can see this is a confirmation one. So this is a B1. So A1 is simply, you can say that what we did in the A. So like we just squeeze. So A, B, whatever the normal position is there, if you squeeze them, like just a spring is there, if you just press the spring. So that if you just press the spring, then you are changing the conformation from B to A. So when you are pressing this spring, then what happened? The distance, the number of nucleotide in that much area, that much space are going to be reduced. So that's why the features, how many number of nucleotides are there per ton, per ring, they are also going to be changed. The diameter is also going to change. The pitch is also going to be changed. So that's why this form are different. So simply what is there, Dna helix pass. So this I'm not reading, just and now come over to the ZDNA. So ZDNA is the reverse of the thing that we did. Instead of pressing down, we are just stretch it. So when you're stretching it a longer distance, so in that unit length, the number of nucleotide will reduced. Okay, so this is but one more thing. Left-handed. So this the hand changes there in your both your in the form of your DNA. Just a moment. Okay. So then types, Next is the types of RNA. So RNA, you all know that the 4 types of the RNA are there, 5 types, and the mRNA, tRNA, rRNA, heterogeneous RNA that is called hnRNA, and small nuclear RNA, snRNA. So these are the different types. Important one are going to be these 3. These are also because they are not involved in the first 3 are going to be involved in your protein synthesis, transcription, and the translation. Later one are mostly part of the control process or the post-transcription process or expression process, how they are going to control them. So structure-wise, these RNA are there, they are single-stranded, but internal double-strand is possible because they are nitrogenous-based. Wherever they found the complementarity, they can make a double-stranded one. So why they do not have a double-stranded, but RNA is not double-stranded? The reason behind RNA is not double-stranded because when RNA is only formed after transcription. Without transcription, RNA can't form, and only one strand is transcribing. That is the reason why RNA remains single-stranded, not the double-stranded. But in many of RNA viruses, RNA is also double-stranded there. So double-stranded RNA genome that you are having, because in that the replication is happening in a way that both the strands are going to be transcribed or synthesized. That's why you have double-stranded there. But as complementarity, I'm saying because of complementarity in the structure of RNA, there are chances are there the loop and hairpin formation. So hairpin or loop, or you can say the loop and stem formation. Wherever you found the complementary, stem is going to form, the pairing is going to happen. Pairing here also in a way that one strand will run in 5' to 3', other is going to run 3' to the 5'. Anti-parallel is going to happen here also. Okay, so then about the structure, what structure about? So mRNA is mainly part of your messenger. It contains the message. So it contains all the machinery which required for the translation. So translation required to attach to the translation machinery that your ribosome. So it have different things. So in case of eukaryotic RNA, have poly-A-tail letter 3'. Then untranslated regions are there, UTRs, 3' UTR at 3' and at the 5' end you are having cap. So cap is nothing, cap is just a modified guanosine. So you are adding this later. So 7-methyl guanosine, if you are adding to that of 5', to that we call the cap. Okay, so 5' UTR is there, then start codon, stop codon, they are going to be the coding sequence. So they are the component of your mRNA. When you talk about the rRNA, rRNA is the RNA component of your ribosome. And ribosome is also, as when I ask you to hear chromosome, as a chromosome in nucleoprotein complex, ribosome is also nucleoprotein complex, means it have both the component DNA and RNA and your proteins. So here we are just referring the RNA component. So both are having respectively size. So different, this RNA like 70 and 80s we are having, it's further large and small unit. Your larger and small unit further have some units that are the 5s subunit and 23s subunits. So with the protein they correspond to the 70s or the 80s. tRNA is the job of the transferring RNA. Transfer RNA, it carry the amino acid and it's carry amino acid, so it help in the reading the message that present on your mRNA. So accordingly its structures are going to be there. Its structure is a bit complex as compared to the other RNA. So it have the 5', at 5' terminal it have the phosphate group, and then it have acceptor arm, that CCA tail, D arm, anticodon arm, T arm, and modified based. These all things, these components are going to be there in your tRNA. So generally we describe 2 types of structures. One is the secondary structure and one is the primary structure and the linear phase. Secondary structure, secondary pairing is going to be there, and then the tertiary structure. In principle, that structure occur in your normal cells. It is going to be tertiary structure. But to understand its structural feature, we describe in case of secondary structure also, so that we can understand what different arms are there, how different pairings are going to be occur. Then later, this secondary structure turn to the tertiary structure. So most important one that I mentioned, it is going to be your 5' is going to be phosphate group. Nothing is going to be there, but at the 3' it is going to be CCA tail. Okay, CCA tail is there at this end. Then they're going to acceptor arm, then T-Psi arm. The CCA tail is the one which is where the amino acid is going to be attached. So that's why you call it the acceptor thing. And the T-Psi loop. So T-Psi is based on, because it have a modified base, it's a psi. That's why you call it T-Psi loop. Variable arm, then anticodon loop. Anticodon loop means this loop contains the anticodon. this the red one, this triplet, the anticodon is going to be there. So this the shape of this anticodon loop is going to give you the reason why the wobbling in the base pair bases is there, why there are more than one codon is there for one amino acid. In the D loop, so this is structure wise. So why DNA evolved as a genetic metal, not the RNA? So this is a simple question. So the reason behind is the between the structure things. So the structure is going to be more stable in case of DNA and less stable in case of RNA. And thing is that the chemical reaction that can occur within the RNA, you can see that the second position oxygen is there. So this second position oxygen is a bit near to the phosphate group, so that a bit near to the 3' OH group. So these can lead to the cross linking. So this cross linking we would like to avoid, because that 3', that phosphate group is there, phosphate group with the 5' one end. So there can be then this structure change can occur. So that's why it's not stable. So each and every time you need an enzyme to break this bond. So that's why we may think that RNA is not a genetic material as compared to DNA. And stability also, DNA is much more stable than the RNA. The sequence of DNA and RNA, what do you know the sequence? So sequence simply means chain, how their letters are arranged from 5' to 3'. So while representing the sequence, whenever you write a sequence, some conventions are there. So you have to follow all these conventions while representing structure of DNA. And these conventions are always you will write sequence in 5' to 3' direction. And you will write only one strand, because second strand is complementary to it. So we won't write the second strand. So what strand we should write? We'll always write the sense strand. So sense strand and anti-sense strand, two strands are going to be there. DNA, gene, genome, so these things are there. Okay, so this is a genetic code. So you must also study what this genetic code is. So a genetic code discovered by the Nittenberg, Maroy, and Rrna. So that's why we are having it. So these 3 base are there. So 3 base means triplet codon. So by this table you can get how this combination can be there. And each triplet is going to be corresponding amino acids that you see. These are phenyl, so some stop codon is also going to be there, which you can see here. So wobbling, as I mentioned, wobbling is there because of the position of the third base in your tRNA. It's a bit away from the codon, so that its location it's a bit tilted. So when it's tilted in a way that G can, if you at the 5' G is there in your anticodon, then it may pair with U and C. So U and C, it means U is G is meant to make 3 hydrogen bond, but its orientation make in a way that it is able to only make 2 hydrogen bond. So 2 hydrogen bond, it means when pairing occur, you can also make pairing with the G. So because of the third location, this wobbling is there. That's why whatever the difference is there, here you can see like for serine there are 4 nucleotides. You can say U, UC, UC, UC. First, second position is common, but third one is a C, A, UC are there. So this is that's why wobbling is there. Okay, so in principle, you don't need all the 64 RNA, 64 tRNA. Even if you have the 32 tRNA, that is more than enough to resolve, to do the job of your transcription, because the wobbling is there only in case of when it is G or U is there at this location. Okay, so the because if A is there, that's not going to change much, but U can do, because they are going to form. They are not contributing to that much hydrogen bond. Whatever their location is, that accepted donor location is playing a critical role in that. So that thing what Manis, I think Yukta was asking, that when you are talking about the mitochondrial genome, there is a universality in the code is there, but this universality is breached in the mitochondria. So the codon that you can see where it is going to be UAG, that is stop codon in normal case, but this stop codon is not the stop codon in the mitochondria. It is for the tryptophan. Simply you can see that those codons which are for arginine in case of normal one, in mitochondria they are going to serve as a stop codon. And those you can see which are isoleucine, that it is going to code for the isoleucine in normal case. They are coding for the methionine in the mitochondria. So that's why universality lost in mitochondrial genome or in the mitochondrial transcription or translation. So this is the how the wobbling is there. So wobbling is so much so that you don't need 32 in case mitochondria. You need just 22 tRNA codon in case of mitochondria. So DNA versus RNA. So these are all thing done. Next thing is the chromosomes. So chromosomes, how it is discovered. The name chroma, color, zoma, body. So this is the thing. What are the different discoverers are there? So again, bit history. So why it is needs? So as I think Yukta was talking on it. So she mentioned that how this chromosome being packaged up. So long DNA is there. So long DNA is just in millimeters. But the size of bacteria is going to be in micrometer. So if you how to accommodate this millimeter range in the micrometer space. So that's why you package in a way that they come into a very lesser space. So that's why here's the different genome size. And if you just start calculating on the basis of genome size, how much the length of DNA in meter or millimeter is going to be, just multiplying by the 0.34 nanometer with the number of nucleotides that you have. Because each nucleotide in your DNA is going to have 0.34 nanometer or 3.4 angstrom. So how DNA is packaged? So in the packaging, you need help of proteins. So protein help in the packaging because your DNA got coiled, your nucleic acid got coiled around your proteins. So whether it is going to be your virus, whether it is going to be your bacteria, whatever different, the packaging, subunit packaging, your DNA is going to be different. Like you know that a nucleocapsid protein is there, so that means that help in the packaging. So that nucleocapsid proteins are going to be there in case of viruses. And so while genome, how it is going to be packaged up. So this is about that. Bacterial genome also going to be packaged up, but the protein which are going to be involved in the bacteria, they are going to be non-histone in nature. They are not histone type. So here you can see HUP is the name of protein. HU protein, HI protein, or unidentified different type proteins are there that play a role in the packaging of your DNA in bacteria. So here simply DNA can be the chromosome term we can use for the bacteria also. So bacterial genome, bacterial chromosome is going to be mostly circular, but it's not always that bacteria is having circular DNA. So when we are saying bacteria have circular DNA, it's not universally true. Some bacteria may have linear DNA also. So here in this, you can say the Agrobacterium tumefaciens, it have a linear DNA and both circular DNA. So both the type of DNA it have. And if you go and the copies wise also, there is not a single copy. Sometimes the multiple copies are also there. So diploidy is also exists in your bacteria also. So if you see Brucella, it have 2 circular DNA. And if you go down the Paracoccus, it has the 3 circular DNA. And so these are the ways that how different types of the DNA are there. So both circular, linear, the possibilities are there. Different type of your. But mostly it is going to be circular only. Few genus are there in which linear is there. Like Borrelia have the linear, and the Agrobacterium have the both the type. Okay, so this is another important thing about the supercoiling. So how the supercoiling is to be. So twisting when you are doubling is there at least to coil around each other. That to the relax. So apoptosis is enzyme which help in the removing the supercoils or adding the supercoils. And whatever the DNA that you are having, natural DNA is negatively supercoiled. So all of us having the negatively supercoiled DNA. So bacteria also have a supercoiled DNA. Eukaryotic genome is also supercoiled. So eukaryotic genome packaging is quite different, as the size is also very large in it. So we have a chromosomes, chromatin, different packaging status in it. So the term chromosome that I asked, I told you when I call the term chromosome, we are referring this X shape. So 2 identical chromatids are there, P arm. And short arm we are calling P arm, and long arm we are calling Q arm. This long and short, P is just peptide, Q is just random Q, and centromere is there in between. So constructing point where the 2 chromatids join together. And DNA molecule is present within your chromosomes. which is wrapped and packaged around proteins, and proteins are mainly histone, and non-histones are also there in them. Okay, so two types of the packaging is there, euchromatin, heterochromatin. That depends on how tightly they are packed. Chromatin structure, how it is going to be packaged. So protein DNA by mass, if you say the by mass protein is going to be 2:1. Means 2 protein is there, 1 DNA is there. So protein is very large amount of protein is involved in the packaging of your DNA. And if you see a second ratio, there's a histone DNA ratio. So 1:1. So 1 histone, 1 DNA is there, but another 1 protein is also there that is going to be non-histone in nature. Okay. So structural unit, if you talk about structural unit, is going to be your nucleosome. That has a structure. Yukta might be Yukta, or who was the second? I'm not remembering. They have explained. So the histone tails, histone is going to be 3 helix stone. So histone structure we may leave. So how this is going to package? So octamer is going to be there. So octamer means H2A, H2B, H3, H24, each of 2. It's called the octamer. And around this octamer it is going to package. DNA is entering and this is exiting. So exit, entry. Between the exit entry we call it's a 1 turn, like this entering and this is exiting here, 1 turn, and this is a second turn, second circle. So totally we can say that not 2 turns are completely. There is 1.7 turns left-handed supercoiled superhelix is there in 1 nucleosome. Okay, so if you talk about how much length of DNA is there, so by average, the octamer that you also call the 4 particle, 446 base pairs are there. By in average in your nucleosome. Okay, so histone H1 is going to serve you as a linker, and they're linking to one exit and entry to nucleosome. They are linking together, so they won't allow in the opening up. The packaging is happen in multiple fold. First fold packaging, second fold packaging. So the 10 nanometer fiber, 30 nanometer fiber, and more thicker fiber that is there. That leads the formation of chromatin and heterochromatin. This you can leave. This you can leave. This. This we can leave. Okay, so this is the just. These are not for you. Okay, so this is the centromere. Another important one. A thing about your chromosomes. So based on the centromere, you can classify your DNA. So about the history of centromere, Theodore Boveri who named it, and Benedon who discovered it. So the centromere, the location you call the metacentric, submetacentric, acrocentric. Meta means exactly in between, middle means both arms are going to be equal. It's almost equal, we can say, if not. So chromosome number 1 and 3 in your human chromosome, they are going to be metacentric. Submetacentric means away from the center. Acrocentric means at the peak. Telocentric means at the terminal. Acrocentric means near to the terminus. P arm is very short. That occur in your chromosome 13, 14, 15, 21, 22 telomere at the terminus. And subtelocentric centromere located closer to the end but far from the acro. Holocentric throughout the chromosome centromere is there. We call the holocentric. That example that holocentric occur in your nematodes, not occur in means we don't have a holocentric chromosomes. We have mainly submetacentric type or acrocentric type. Okay, so here you can see different types of the chromosomes. So by just seeing, you can tell that which one is of what type, whether it is of acrocentric or metacentric. So you can see this is going to be submetacentric. This is going to be metacentric. Exactly into the center, both arms are going to be equal. And this one is going to, you can see this is smaller one, Y chromosome type. This is your acrocentric. And this one is also acrocentric. Exactly near the terminus is there. So telocentric is this. This is subtelocentric. So these are different types of chromosomes that are present within you. So arranging them on their size, we call them the. Okay, centromere is a site of attachment that help in your segregation. Okay, another important component of your chromosome in the structure, you need to describe of the telomere. What telomere is? Telomere, as mentioned, it is a tetrad, four-stranded DNA is there. So telomere, one of the beauty with the telomere is that telomere is not replicating one. Telomere do not replicate when the replication occur. Telomere is being synthesized not artificially, naturally by the enzyme that is or later after the replication. That name of the enzyme is telomerase that synthesize it. And telomere mainly have a sequence that T, A, G, G, G. That's as we're talking about the repeats. So this sequence keep on repeating and repeating around 2,500 times in human. So this is the one. And at the last, it is going to have a single stranded overhang. So that is going to be like GGTT and GGGT. The sequence is there at the last that leads to formation of an overhang. Okay, so this is a structure of telomere. G tailing is there. Okay, so this G tail we call the telomere. And this length of telomere is very important. This length of telomere also can tell you the age, because as we keep growing on, length of telomeres keep on decreasing, and when we are near to our deathbed, size of telomere become very small that it can't sustain our life and we are going to die. Okay, so this is also a genetic clock. Telomere play a role of your genetic clock, how it and it determine your age. So why its length is going to be short down? Because it is not replicate, it is synthesized, and synthesized by the telomerase activity. So telomerase activity keep on coming down, so it won't synthesize the length as it's required at the time of your birth. Okay, so role it stabilize and seal the chromosome ends. So it's its role. So sealing is going to be the tetrad structure that happened. That we can say it is going to be there. So this the four-stranded thing is there. So this loop type of structure forms at this end. So that leads the formation at sealing it. So this end is not available for any type of the linking with other than, so no circularization occur. A partial circle form within the telomere. Okay, so other useful feature in your DNA is that large chromosome. So that large chromosome is on the lamp brush chromosome. So as the brush is there, it have brushy-like of appearance. That we call it as a lamp brush. Your lamp cleaning brush is there. It's unusual chromosome and found extended and allows individual loci. So it found not found in everyone. We don't have it, but it mainly found in amphibians. And so the main role of why they are doing, because they are found where they need excessive transcription. Keep on protein expression is very high in those chromosomes where it found. So it found in your amphibians like frog. Length is 5 to 6 millimeters. So this much length is so big that you can see by your naked eye also. So you call it a large chromosome. Second time is the Paulietne chromosomes. So Paulietne chromosomes also the role of same. It required for when the high protein requirement is there. So high level of transcription is there. So difference between the Paulietne and the lamp brush is all are a single stranded one. Means like only normal type of the chromosomes are there, but except the size, everything is going to be same. But in Paulietne chromosomes, there are multiple chromatids are there. Giant chromosome is produced by successive replication. So, but they do not separate. All the chromatids are attached at centromere. So diploid pair, it replicate but do not separate, remain attached in their extended form, and they relatively large and usual. So when they are keep on multiple structures are there, multiple chromatids are there, they're not separating. That's why you can see them. They also required multiple strands when you need expanded gene expression. More the strands are going to be there, they can transcribe more. So more RNA you can produce. Okay, so this bending pattern is there, that's called the G band. So G band, G stands for GIMSA. When you stay in your chromosome with the GIMSA, then it will give you bending appearance. So they will call the bands, G bands. Okay, so this about the chromosomes, and now we move to the last part of this module, that is about the organelle chromosome, that is the mitochondria and plastid. So I will share you one thing here that who talked? Yukta talked, I think. Who other than Yukta talked? Nandini, Yukta, and नंदिनी युक्ता और किसकी थी तीसरे?
Saiba.
Saiba, yes. Saiba, Nandini, Yukta. Okay, so one of them talked about the mitochondrial DNA. So when we're talking about this nuclear DNA and mitochondria, we must talk about their origin, how they are originated. In principle, they are a parasite which are residing, not you can say they are a symbiotic parasite. They are residing within us. They are independent one, but they independent organism. But association is so symbiotic that we can't live without mitochondria, and mitochondria can't live without us. Are also. Okay, so they are derived from endosymbiotic bacteria. So whatever the mitochondria we are having, it is a bacterial DNA. Bacterial genome is there in it, so that's why mitochondrial DNA is circular in one. Endosymbiotic bacteria, free-living prokaryote that invaded in the ancestral eukaryotic cells and established mutual beneficial relationship. So that's why they stay there. And when you associate a mutual relationship, then what you start doing that some of job, if you share, you do my this job, I will do your this job. So some function it loses. So it loses its independent existence. And our cell also, eukaryotic cell also loses its independent existence. None of these two can exist independently, and now they are occurring in a way that we call both as a single entity. Okay, we not consider them as separate. So a nuclear mitochondrial DNA transposed to nucleus, so ribosomal RNA. So this thing. So thing is that now if you talk about the ancestry, so ancestry in case of the mitochondria and ribosome both share different ancestry. Ribosome share the ancestry with the cyanobacteria, whereas the mitochondria share ancestry with the agrobacterium or the rhizobium. Okay, so this is the how it is going to occur. So this is the genome analysis. If you study genome mapping, if you check. So mitochondria originate from the archaea or eukaryote, and plastid have origin with the endosymbiotic eukaryote. And the protobacterium, then the protomitochondrion, then the protomitochondrion to mitochondria heterogenome. So how this ancestrally they forms. So organelle genome, if you talk about their genome size, how they are going to be. Their genome size very depend upon the plant to plant, or say animal to animal. So if you talk about the plant plastid, it is going to 150 kilo base pair circle. Plant mitochondria, it is 150 to 2000 kilo base pair multipartite. Multipartite means more than one particle is going to be there, one more than there in the mitochondria. Human one is going to be 17 kb, very smaller. Plant is going to be larger. So only very few number of genes are there, 37 genes, 22 tRNA, 2 RNA, 13 oxidative phosphorylation. These genes are there. Yeast mitochondria, it is going to be 75 kilobase pair long, so it is bigger than the human one. And amoeba, again a parasitic, its bacteria, its mitochondria is again 10 to 100 kilobase pair. So how in the evolutionary history, how in individual organisms, how they establish themselves, that leads to how much genome size they require, that make the mitochondria of its size. Okay, so that's it about your module 1. So with this we close our module 1, and I will share you some of these slides which are of your importance, and also the slides that is Nandini, Saiba and Yukta shared. They will also share with you. Have you uploaded these slides? Have you uploaded your slides?
Yes, sir.
So only 2 of you have uploaded. Who didn't do it? किसने नहीं की? Nandini done. Sahiba. Sahiba is absent.
Sir, she is facing some network issues.
Okay, ask Sahiba to upload her assignment immediately.
Okay.
Okay, if you have any query, you can ask, because this module we run very fast. Quickly, I ended up because 4 of us were talking about same things. So that's why I didn't, because you already have studied these things in your previous semester also. But as a ceremonial, we have to talk. At least we have to revise your concept. just being fresh then, because the questions are definitely going to be there from this module also. Okay, if no question, then please turn on your camera while I call your name. Adish. Adish.
Present, sir.
Sir.
You were absent in your first session. Why? Why didn't you join in the morning session?
Sir, I have network connection this one.
Ayushi.
Present, sir.
Ishita. जानवी, मनस्विनी, नम्रत, देवांशी कैमरा भी खोल दिया करो भाई.
Sir, my camera is on.
अभी तक तो दिखे नहीं भाई देवांशी का. Only Manaswini is regular, except Manaswini Ishita. So if you student I name Manaswini Ishita, they turn on their camera regularly. Once you call the name, they turn it on even at the start. But others are shying in turning on the camera. And one Nandini never turn on the camera. Namrita, Nandini, Nehal, Rupal, Saiba. Earlier Saiba was
Present,
also
sir.
turning on the camera. Now Saiba also.
Sir, am I visible now?
Yes, Devanshi. You are.
Sir, am I visible?
Okay, Shakti is also one who don't like to turn on camera. Shivani, Tanvi, Tridev, Vivek, Adish Bhavya. Okay, so thank you all. Stay safe, stay happy. So we will join back next week. Till then, enjoy your weekend.
Thank you,
Get
sir.
ready for your test. Okay, so next session we will devote for discussion. If any query you have, then after that, I think on Tuesday we will have test. Okay, is it fine? And on Tuesday itself, I will also I will decide who is going to present on which date and what is going to be their presentation topic. Okay, so
जिन्होंने पीपीटी बना लिया उनका क्या फिर?
उनका कुछ नहीं, दोबारा बनाना है. Okay, so thank you all. Stay safe, stay happy.
Thank you, sir.
Thank you, sir.